Cell is the structural and functional unit of all organisms. You can imagine cell as a factory which has a controlling center, that is nucleus. Then there is source of energy also, that is mitochondria, synthetic units as well. Then there is a full machinery for transporting the units synthesized to various places. So basically, the operating aspects of a cell are the organelles. Now, various organs have cells with these organelles, which are more or less same in almost all the cells. But there are certain cells with specialized functions in which these organelles may vary. For example, a cell which needs to use lot of energy has more mitochondria. On the other hand, a cell which is synthesizing lot of proteins will have more ribosomes, endoplasmic reticulum, and Golgi complex. Then there are cells which do not divide and they do not have nucleus. But fundamentally, the function of any organ is because of its cells and its organelles. So let us see the structure and functions of mammalian cell. Well, generally, cell is depicted as a spherical structure which is having different organelles. But that is not true. Cells have various size and shapes. For example, neuron. Neuron is an elongated cell. Similarly, there is muscle fiber, which is an elongated cell. Anyways, with this, let us see the various components of cell, that is the cell membrane and its organelles. So, the cell membrane or plasma membrane is mainly phospholipids, which is basically phosphatidylcholine, phosphatidylserine, phosphatidylethanolamine, and there are other lipids as well, such as cholesterol. But cell membrane is not only lipids, it consists of a lot of proteins as well. In fact, proteins form 50% of mass of cell membrane. So here is shown a cell membrane, you see, which this yellow part is basically lipids. Then there is a cholesterol. Then embedded in this cell membrane are various proteins. So there are proteins which are traversing the entire cell membrane, right? So one part of the protein is on the intracellular side, while the other part of the protein is on the extracellular side. So this is an integral protein which traverses the cell membrane. Then there are peripheral proteins which are present on either side of the membrane. So these proteins embedded in the membrane are basically of two types, integral proteins and peripheral proteins. Now these peripheral proteins are attached to the lipid membrane by two methods. Either there can be a GPI anchor or there can be a lipid chain, what is known as lipidated protein. So basically, this lipid chain is inserted into the membrane, and on other side of this lipid chain there is the protein. So this is example of lipidated protein. Similarly, GPI linked protein will have GPI anchors which are inserted into the membrane. So what are the functions of the proteins which are present on the plasma membrane? Well, they act as cell adhesion molecules. So by the use of these proteins, the cells interact with each other, make cell-cell contacts, and they also interact with the extracellular matrix. So what is formed is cell extracellular matrix junctions. Then these proteins also help in transport across the cell membrane. So they act as pumps, carriers, ion channels. They also act as receptors. So there may be certain chemicals which are present in circulation, and these chemicals can bind with these plasma proteins. Plus, they can act as enzymes as well. So there is an enzyme like adenylate cyclase, which is present on the membrane itself, right? It is attached to the membrane and helps in second messenger mechanisms. So the proteins present on the cell membrane perform various functions. Now, the cell membrane gives the cells a polarity. Polarity means the apical part of the cell membrane differs from the basolateral part of the cell membrane. Now, the proteins which are present on the cell membrane, their arrangement on the cell membrane provides the cell a polarity. What does this mean? Actually, the apical part of the cell membrane is different from the basolateral part of the cell membrane. So if we see, if this is the apical part of the cell membrane, right, and you see this entire other aspect, this is the basolateral part of the cell membrane. That is why it is known as basolateral. So lateral part of the cell membrane and basal part of the cell membrane is taken together. Apical part of the cell membrane faces towards the lumen. Now, at the junction of apical membrane and the basolateral membrane, we have tight junctions. So what happens? The proteins which are present on the lateral and basolateral side of the membrane are different from the proteins which are present on the apical membrane. And because of these tight junction, these proteins cannot move, right? So it is separating the apical compartment from the basolateral compartment. And it is because of this polarity that cells can perform their functions properly. Because you see, if there are certain transport proteins which are present on the apical membrane and they move and they come to the basolateral side, then it will not be possible for the cells to transport the substance from the luminal side into the cells and then from the cells into the basolateral side. So this apical polarity is very important for the functioning of the cells. So what are the functions of cell membrane? Obviously, the proteins are performing various functions. Apart from this, cell membrane actually acts like a selective barrier. So it is not allowing substances to cross freely from one side to the other side. But it is allowing some substances to pass through it. So that is why the term selective barrier. Secondly, because of the proteins, whichever substances need to be transported from one side to the other side, they can be easily transported. So it is acting as a barrier on one hand and it is causing transport of substances on the other hand. Then it is receiving the information because there are proteins which are present on the cell membrane which can bind with chemicals, isn't it? So these chemicals form a way of communication from cell to cell. So information is received by the cells. Then there is transduction of information. So suppose a hormone binds to the receptor which is present on the cell membrane. Then on the cell membrane, this information is then transduced to chemicals inside the cells. Right, what is known as second messenger system. So there is reception of information, there is transduction of information so that further changes can be made in the cell. Then cell membrane helps cells to communicate with each other. So there are cell-cell junctions, cell extracellular matrix junctions. So this cell membrane is very important for communication as well, which is a very important property of multicellular organisms. With this, let's come to the various organelles of cell. So first organelle is mitochondria, very popularly known as the powerhouse of the cell because it is the site where ATP is generated. Now these mitochondria are round or oval in shape and their number varies from less than 100 to greater than 300, and in some cells they can be up to 1000 mitochondria. The mitochondria are bounded by a double membrane. So you see this is the inner membrane and this is the outer membrane. Inner membrane, you see, it is thrown into folds and these folds are known as crista. This mitochondria is bound by double membrane. So this is the inner membrane and this is the outer membrane. Inner membrane, you see, it is thrown into folds which is known as cristae, right? And there are various proteins which are present in these folds. And remember that wherever folds are present, it actually increases the surface area. So obviously there will be a lot of proteins present on these folds. Then outer membrane is actually permeable to water. So there are two aqueous compartments in the mitochondria. One bound by inner membrane, the inside one, which is the matrix, and the outer one bound by inner membrane and outer membrane. This matrix contains the DNA, ribosomes, and enzymes which are required for replication of mitochondria. Fine. So what are the functions of mitochondria? Mitochondria is the powerhouse of the cell, which I think everybody knows, but it also has various other functions. It has role in apoptosis. There is a protein, cytochrome C, which is present in between the two membranes of the mitochondria, and when it is released from the mitochondria, it activates the apoptosis. Then it has various metabolic functions also, and it is important for beta oxidation of fatty acids. Final steps of the synthesis of steroid hormone occur in mitochondria, and it is also a part of urea and heme synthesis pathways. Plus, it also regulates intracellular calcium concentration. So next time, if anybody asks you function of mitochondria, just don't say only powerhouse of the cell. It has various other functions as well. Next organelle is lysosome, where degradation of waste products takes place. So for degradation to occur, there should be certain enzymes which hydrolyze the products. So yes, this lysosome contains granules with hydrolytic enzymes. Also, these hydrolytic enzymes actually act in acidic environment, and there is a pump, hydrogen ATPase pump, which is present on the membrane of lysosomes, which maintains acidic environment in the lysosomes. If you see, it is kind of protective, that if by chance these hydrolytic enzymes are released into the cytoplasm, then they will not be active, since these enzymes act only in acidic environment, right? So their function is restricted to inside the membrane. Now, some cells have special lysosomes, like in WBCs. The granules of WBCs are basically lysosomes. Then there is acrosome in a sperm, which also has hydrolytic enzymes. Now, this lysosome basically is pinched from Golgi apparatus, which is basically a primary lysosome. The waste products which are taken from the surroundings, or any substance which needs to be degraded is taken from the surrounding by endocytosis. It is basically held in a phagosome, which is basically enclosement of the waste product by a membrane. Now this phagosome, when it fuses with the primary lysosome, it forms the secondary lysosome. The degradation of the waste products occurs in the secondary lysosome by the release of the hydrolytic enzymes from the granules which act there, and the products which are left, that is known as residual body. And finally, this residual body is released again to the surrounding by means of exocytosis. So there is discharge of contents outside again once the degradation has taken place. Next organelle is ribosomes. Ribosomes are the protein factories, and they are of two types. Either they are present on the endoplasmic reticulum membrane, or there are free ribosomes. The ones which are present on the endoplasmic reticulum membrane synthesize transmembrane proteins, secreted proteins, and proteins stored in lysosomes. On the other hand, the ribosomes which are free, they form cytoplasmic proteins, proteins found in mitochondria and peroxisomes. So these ribosomes are basically protein factories, but different types of proteins are synthesized in different ribosomes. The ones which are present on endoplasmic reticulum membrane synthesize transmembrane proteins, secreted proteins and the ones which are present in the lysosomes, while the free ribosomes synthesize the cytoplasmic proteins. So, the other organelle, endoplasmic reticulum, is also of two types. The one which is studded with ribosomes, where ribosomes are attached, is known as rough endoplasmic reticulum. And you see here it is basically in continuity with the nucleus. Okay, so the mRNA from nucleus moves outside and goes to the ribosomes, which synthesize the proteins. So these are the rough endoplasmic reticulum, and as the diagram shows, basically they are connected sac-like and tubular structures which are studded with the ribosomes. Now once the ribosome synthesizes the proteins move into the endoplasmic reticulum, and the initial folding of the chain of protein occurs in the rough endoplasmic reticulum. So there is folding of the polypeptide chains, plus there is some modification of the proteins also going on, what is known as post-translational modification, right? So understanding the path, how it is occurring, mRNA is coming out, protein is being synthesized by ribosomes, and then the protein chain which is forming, it is folded in the rough endoplasmic reticulum, and there is post-translational modification of the protein also going on in the rough endoplasmic reticulum. The next one is a smooth endoplasmic reticulum. Obviously, as the name suggests, there is no ribosomes. Okay. And this endoplasmic reticulum is important for the re-esterification of fats, for the steroid synthesis, for the detoxification of chemicals, and it also acts as calcium store in case of sarcoplasm, right? So there is sarcoplasmic reticulum which acts as a calcium store. So rough endoplasmic reticulum has a different function. It is studded with ribosomes and it is causing the folding of the polypeptide chain. On the other hand, smooth endoplasmic reticulum, it has different functions. Fine. Now, once the proteins are synthesized and folded in the endoplasmic reticulum, they move from there in vesicles. And this vesicle actually goes and fuses with another organelle, that is the Golgi complex. So this Golgi complex has two different faces. One which faces towards the endoplasmic reticulum, which is known as cis phase, and the other side is known as the trans phase. So the vesicle which is containing the protein comes and fuses with the cis phase of the Golgi complex. Now in the Golgi complex, the proteins are concentrated. There is some further modification going on, that is known as the processing, and they are packed and they are labeled with some address, because there will be different proteins which have to go at different places. Some have to be secreted, some have to go within the lysosomes. So it's kind that the protein is labeled with an address, right? So from the trans vesicle, you see the vesicle comes out with the labeled protein and it goes into the targeted place. So these are the functions of Golgi complex. Next organelle is peroxisomes. Peroxisomes are ovoid structures present in hepatocytes and proximal convoluted tubules in kidney, and they contain oxidative enzymes. These organelles are very important for protection from the free radicals. So they basically cause degradation of the peroxide. This is one of their most important function. Additionally, they cause synthesis of the bile acids, important for cholesterol synthesis and fatty acid oxidation. Then, next organelle is nucleus, which is the storehouse of genetic information. If we see, this nucleus consists of envelope, right? So there is a double membrane in this envelope, inner membrane and outer membrane. And this membrane is interrupted in between. So there is nuclear pore, because some substances move into the nucleus and out of the nucleus via this nuclear pore. Then you see the membrane is actually continuous with the endoplasmic reticulum. As I said earlier that if mRNA comes out, it has to reach to the ribosomes for the synthesis of the proteins. Okay. Inside the nucleus, what we see is chromatin, which is condensed DNA, and there is also nucleolus. So this nucleolus is basically collection of granules and it is made up of ribonucleoprotein. See, if ribosomes are synthesizing the protein, then what is synthesizing the ribonucleoproteins? Well, it is this nucleolus which is made up of ribonucleoprotein and it does not has a membrane. It is just the collection of granules. Finally, we have certain cytoskeleton elements also in the cell. So there are basically three types of cytoskeletal elements. There are microfilaments, intermediate filaments, and microtubules. So each of these cytoskeletal elements have different function. So if we see the microtubule, it basically consists of some repeating units. So you see that there are synthesizing units which are two different protein substances, alpha tubulin and beta tubulin. So let's see one filament of microtubule. In one filament we have one alpha tubulin and one beta tubulin, and then again there is a repetition of this. So this is one again alpha tubulin, beta tubulin. So this entire filament is the protofilament, and many of these protofilaments, that is 13, form the microtubules. Now these microtubules can grow at one end, which is known as the plus end, and they decrease in size from the other end, which is known as the minus end. So more alpha tubulin, beta tubulin units bind to this end, while from the other end they get dissociated. So that's how there can be growth of microtubules on one side, while it degrades from the other side. Now this microtubule has various functions. First of all, it is important for vesicular transport. There are certain molecular motors which carry substances from one end of the microtubule to the other end. So examples of these molecular motors are kinesin. So kinesin is a molecular motor which moves from the negative end to the positive end of the microtubule. On the other hand, there is dynein, which carries the substances from positive end to the negative end of the microtubule. So this transport from the negative to the positive end by molecular motor kinesin is basically occurring in anterograde direction, from negative to positive. The opposite direction is known as retrograde transport. Second function of a microtubule is formation of the mitotic spindle in case of the dividing cells, which is happening in metaphase. Then it also forms the cilia. So microtubules important for vesicular transport, formation of mitotic spindle, and for cilia. Next type of cytoskeletal element is actin filament. So actin filament is basically formed from G-actin, as microtubule was formed from alpha tubulin and beta tubulin. Here this microfilaments is formed from, so a number of G-actin monomers come together to form the F-actin. So this is important for muscle contraction, formation of cell-cell junctions, that is the adhesion belt, and cell extracellular matrix junction, that is the focal adhesion. Then like microtubules, they also act as tracks for molecular motors. Plus, they are important in the formation of microvilli and lamellipodia. So these are the various functions of microfilaments. Next type of cytoskeleton filament is intermediate filament, and this is a kind of a stable cytoskeletal filament. It is static, doesn't change much, unlike that of microtubules and microfilaments, and it provides structural support to the cells. So in summary, we saw the characteristic of the cell membrane of the cell and the structure and functions of various organelles, that is the endoplasmic reticulum, Golgi apparatus, lysosomes, ribosomes, mitochondria, nucleus, and then we also saw about the cytoskeletal elements which are present in the cells, that is the microtubules, microfilaments, and intermediate filaments.